Hi everyone, and welcome to our video on some exam questions related to Unit 4 DNA, RNA and Protein Synthesis. Our first question is from the 2016 AQA AS Biology Paper 1 and states, Table 1 shows features of a bacterium and the HIV particle. Complete Table 1 by putting a tick where a feature is present. Okay, so we've got the feature in one column. We've got the bacterium if it contains these features, and then we've got the HIV particle if the particle contains the following features. And we just need to place a tick in the features that each of these contain. So if we start off with RNA, RNA is contained within bacterium, so we would just place a tick in there, and it is present in HIV as well. Okay, now cell wall is present in bacteriums. They have a murine cell wall. However, it's not present in HIV, as HIV contains the attachment proteins which they then use to bind to the receptors on body cells. Okay, enzyme molecules, yes, bacteria does contain enzyme molecules. And HIV does as well, because HIV contains reverse transcriptase, okay, which allows the DNA to be replicated in the HIV particle, allows the particle to be replicated and spread. Okay, so that is present in both of those. And finally, a capsid. Bacteriums don't have capsids, however the HIV particle does have a capsid. Okay, and if we have a look at the mark scheme, the mark scheme says just that. Okay, and you would get 1 mark for each correct vertical column. Okay, and that's why it's worth 2 marks. For each vertical column that you've got correct, you will get 1 mark. Okay, part 2 of this question says, when HIV infects a human cell, the following events occur. A single-stranded length of HIV DNA is made. The human cell then makes a complementary strand to the HIV DNA. The complementary strand is made in the same way as a new complementary strand is made during semi-conservative replication of human DNA. Describe how the complementary strand of HIV DNA is made, and it's worth 3 marks. Okay, so this is basically relating back to the nucleic acids unit, which you should have done as one of the first units. And it's telling you that the complementary strand is made in the same way as a new complementary strand is made during semi-conservative replication. Okay, so that means it will use the same enzyme when making this complementary strand, which is DNA polymerase. Okay, so we've got DNA polymerase. And remember, the hydrogen bonds come together between complementary nucleotides. Okay, so the hydrogen bonds occur between complementary nucleotides, or you can say bases. Okay, and if you're saying bases, just remember to write that A pairs with T and C pairs with G. So adenine pairs with thymine and cytosine pairs with guanine. Okay, and once these nucleotides have formed, the backbone of the complementary strand will form. And how does that form? Well, it's because of the phosphodiester bonds. Okay, so the nucleotides join together with each other to form the backbone of the strand. And what bond is formed? It's a phosphodiester bond. Okay, So that's what I would write for 3 marks. If we have a look at the mark scheme, it says complementary nucleotides / base pairs or A to T and C to G. Okay, and as you can see in our answer, we have both of them. We've written A to C and C to G, and we've written complementary nucleotides and bases. Okay, DNA polymerase is required in your answer because it says in the question that it's made in the same way as the new complementary strand in semi-conservative replication of human DNA. And the nucleotides join together, but we have stated that they form via a phosphodiester bond. Okay, so this is basically a question which overlaps with a previous unit. That's why the mark scheme is presented in this way. Okay, it says ignore DNA polymerase forms base pairs nucleotide pairs. If clear writing wrote answer about DNA replication, you will get 2 marks because this is slightly relating to the immunity unit and a past unit on nucleic acid. If you've not used any knowledge of the immunity unit, then you will only get 2 marks. Okay, final question. It says contrast the structures of DNA and mRNA molecules and give 3 differences. Okay, now the most obvious difference that you can give here is that DNA is double stranded, so double stranded, and you need that, and mRNA is single stranded. Okay, whenever you are comparing or contrasting, you need this contrasting word. It could be and, it could be while, anything which contrasts the 2 properties. Okay, or else you won't just get the mark. Okay, now mRNA is a type of RNA. Okay, there are 3 types of RNA that you need to know: mRNA, tRNA, and rRNA. Okay, so we are speaking in RNA terms through this. Okay, so we can also say that DNA is long while RNA is shorter. Okay, what we can also say is that DNA contains thymine while RNA contains uracil. Okay, we are just comparing the structures of DNA and mRNA molecules. Okay, if we have a look at what the mark scheme says, it says that DNA is double stranded or a double helix, and you need that and in all of these cases. As it says over here, contrast requires both parts of the statement. If you don't state the and, you are not stating both parts of the statement. Okay, mRNA is single stranded, DNA is very long, and RNA is short. Thymine in DNA, uracil in RNA. Deoxyribose you could have had as well in DNA, and ribose in RNA. DNA has base pairings while mRNA doesn't. DNA has hydrogen bonding whereas mRNA doesn't. DNA has introns or non-coding sequences whereas mRNA doesn't. Okay, remember they are all spliced out. However, you can't state that the mark scheme says ignore reference to splicing, because splicing comes in when you're talking about transcription and translation. Okay, and that is it for this video. Thanks for watching. I hope you liked it, and one last thing, please subscribe, hit the like button and the notification bell.